¿no? Empezamos ya por la primera presentación, que es a cargo de Jakub Czornacki. Él es un research scientist del Instituto de Irsicasa, del Instituto de Investigación del SIDA, y también es manager de la facilidad de la Microscopy Facility del IGTP del Instituto Germán Trias y Pujol. Él hizo su doctorado en la Universidad de Melbourne y luego hizo dos postdoctorados en la Universidad de Heidelberg y luego en la Universidad de Oxford, y fue en 2018 que se integró en Irsicasa, y ha estado trabajando mucho en la microscopía convencional, pero así también de la Super Resolution Fluorescence Microscopy, que es lo que nos hablará hoy mismo. Así que muchas gracias.
All right. Thank you very much. First of all, I'd like to thank all the organizers for allowing me to present my talk. This is, of course, more basic research talk, so I'm going to take you a little bit of the story of super-resolution microscopy. So, without any further ado, what my presentation is going to look like is I'm going to first give a bit of an overview into general principles of super-resolution microscopy, what it is, without going into too much details, basically just to appreciate the key message of what super-resolution microscopy is and what are its advantages. Then I'm going to highlight a little bit about super-resolution microscopy research in HIV field in general, and then I'll take you on a little bit of a story of how I personally was involved in the field and what sort of research and studies I have conducted over the years, which contributed to this field, and how I found it to be actually very, an actually amazing journey, at least personally for me. So, overview of the super-resolution microscopy techniques, and I'm sure all of you are aware that fluorescence microscopy is a really invaluable tool of scientific research. Basically, seeing is believing, and all of us have seen amazing images of microscopy. So fluorescence microscopy, of course, the advantages are it's fast technique, relatively, no need for harsh chemical treatments, like, for example, in electron microscopy. Very importantly, the potential for live cell microscopy is there, so we can study things in much more relevant context. And of course, the high throughput potential for tagging of multiple targets. So there is a lot of advantages of using this type of techniques of fluorescence microscopy, but classically, at least, one of the main disadvantages, at least when concerned with virology field, was its resolution. And normally, fluorescence microscopy resolution limit for a conventional fluorescence microscope like confocal or even a wide field microscope is about 250 nanometers across when we're looking at XY or focal plane, or 600 nanometers along the optical axis. And the causes for this are basically due to the wavelength, the nature of the light and interference that it produces, and also imperfect fluorescence light collection. So if you look at the bottom, any object that is, let's say, hypothetical object of 40 nanometers would appear to be in size of approximately 250 nanometers, depending on the resolution of the microscope. And of course, in the virology field, where viruses are generally 100 nanometers, HIV being 140 nanometers, this is unacceptable, and normally you could not study viral detail or molecular detail using fluorescence microscopy. However, about, let's say, already 10, 15 years ago, super-resolution microscopy came into the field. And basically, this was a result of many researchers thinking how to cheat this fundamental limit. And basically, over the last 10 years, a whole lot of acronyms showed up in the field. And you don't have to remember all of them, but basically, multiple methods for super-resolution microscopy were designed. We have a STED microscopy, SIM, super single molecule localization microscopy. Also, more recently, MinFlux techniques. And well, I don't expect you to remember all of those, but the bottom line was to convert images like this, where you can see at the bottom left the HIV imaged with the resolution limited microscope, or nuclear probes, giving a blurry image. So to convert images like this into images like this, where basically, instead of just having a blurred individual virus, you can actually start seeing subviral structures within the virus. So at the bottom left, you can actually see the distribution of end molecules on a virus surface, whereas for nuclear pores, you can start seeing actually the complex structures that those nuclear pores are. And this is what I would call a current generation of super-resolution microscopy. This has actually been available from about 2010. So this is not actually already a new technique. This technique actually is kind of well established, and more microscopes actually come with this set of techniques by default built in. So any new microscope that is purchased right now, there are a good chance is that it will include a super resolution modality. So it's no longer confined to physicist labs or a small subset of groups. Because right now already on the market we have already coming to the market, I would call a second generation of techniques which offer a fluorescence microscopy resolution of around 1 or 2 nanometers. So we're talking about a real molecular resolution in which the orientation of your fluorescent probe makes a difference. And those techniques are already also coming to the market as well, becoming commercially available. So there is actually there has been actually an immense amount of research and innovation since the classical days of confocal. Definitely the field has moved on quite a lot since then. So, just to move on, give a little bit of a brief overview of, well, how HIV replication cycle has been studied over the years with the super resolution microscopies. So, this is the part of the review that I did in 2018, and there has been actually an immense amount of studies through HIV, and I want to argue that HIV is most studied pathogen by super resolution microscopy. So people have tackled assembly and release, virus architecture, virus entry, as well as post-entry releases. However, to date, when I look at even at the review, and to date, this has been confined to a relatively small number of groups, research groups, which took advantage of this technique. Nonetheless, actually, existing studies have already revealed a previously unknown mechanism influencing HIV assembly and entry. And this revealed actually to me and to everyone in the field actually, these studies revealed enormous potential of these techniques for studying virus and virus cell interactions. And this is despite the criticism that often was leveled on these techniques. Oh, you're just studying the same thing that was known before, you're just applying a fancy new microscopy to it, and you're not going to find out anything else beside a high resolution image. That actually is not correct. We found a new layer of complexity that was basically hiding beneath what was already known. And of course, HIV was studied quite a lot because it is the wealth of reagents, the wealth of previous research that was done in this field has made it actually quite easy to then apply the new techniques to already existing body of like I said, reagents and already existing fluorescence microscopy techniques. However, like I said, it was still confined to a relatively small number of groups. So I just want to highlight since then a few studies that have been done since in the early 2020s. Right now, people are developing cost-effective and cheap solutions for studying in live cell imaging HIV transfer at virological synapses. This is all in super resolution that I showed before. The entire largely complete HIV capsids have been imaged inside a nuclei. This is also quite a new finding since, as of recently, everyone was thinking that HIV capsids disassemble in perinuclear region, and this study actually contributes to the findings that actually the HIV capsids disassemble inside the nucleus, which is a very important finding in its own right. Of course, then there have been studies like imaging of the CD4 clustering upon HIV binding, and also quite recently, they're looking at antiviral factors, which also seem to affect the ENF clusters and interrupt viral fusion. And this leads me to the next topic, ENF clusters. As you can see, the few of these last studies which are highlighted here talk about ENF clustering. And actually, ENF clustering is one of those novel findings that I contributed to the discovery of the importance of ENF clustering on the virus surface and how does it affect the virus replication cycle. So this brings me conveniently to my contribution to the field, and the first study that I want to briefly go through is looking at HIV ENF distribution during the virus assembly and entry. That is already quite an old story, but it actually is part of the nice chronological process of discovery which I went through. So, to go through a few basic information about this, at the time, well, it was not much was known about ENF incorporation or what is the role of ENF or ENF distribution in viral entry. The idea was that HIV incorporates a very small amount of ENF that is much lower than theoretically be possible. And ENF incorporation is influenced by these interactions in the main domain of GAG. And actually, it was known at the time that ENF distribution may be important for the HIV entry, as at least in silico studies, several NF trimers were necessary to be found to be necessary for fusion. So the bottom line was that NF distribution may be influenced by its interactions with GAG and thus affect the entry competence of the virus. But up until then, it was not possible to study the NF distribution directly on the virus. So the aims were to, well, precisely do that. Look at how can we visualize NF via superposition microscopy, and to establish whether GAG processing has any effect on it, and to observe any rearrangements that this NF might undergo when it interacts with the cell. So this is where we studied, this is a snapshot of a study where we basically look at immature and mature virus particles using a superposition microscopy. And basically over here, instead of a diffraction limited spot that I showed before, we're able to visualize individual virus particles, which are shown in green, with its own defined NF distribution pattern that is actually here on orange. And we defined several classes of NF distribution and found out that immature and mature particles differ quite significantly between the NF distribution pattern, with a much more clustering, as shown here, seen in mature virus particle versus the immature virus particle. Then we also looked at how those NF clusters colocalize with CD4 receptors. So here was an image of a cell and a virus on the surface of the cell. And we basically, once again, went down to the level of individual virus particles, and we visualized how the clusters of NF interact with CD4, which is actually in this case a blue, and how does the effect of the virus being immature or mature, or missing the portion of the NF affects this clustering. Without too much details on the study, we found out that the HIV maturation actually acts as a inside-out signaling, which signals the NF to recluster as the virus matures, and the NF clustering was actually was crucial to be important for efficient virus entry. And this is actually important given how few NF molecules are on the virus surface. So then, moving on to the HIV surface mobility, a next study. So, this, we wanted then to find out what are the mechanisms which facilitate this change from multi-cluster distribution to a single cluster distribution. And one of the clues was to look at the NF mobility changes, how dynamically the virus changes. And here we had to, let's say, kick up the microscopy a notch a little bit and go into, measure the mobility on the virus surface and establish whether virus maturation alters this. But this we had to study with, well, basically even more advanced microscopy technique called fluorescence correlation spectroscopy. And basically what this technique does is you sacrifice a bit of imaging capability of microscopes, but they gain capability to look at things dynamically. So basically you can measure average transit time of molecules across the field of view, and this allows you to get the molecular diffusion coefficient and molecular mobility of the molecules which have been tagged with fluorescence microscopy. And then combining this with a superposition microscopy, we can image the mobility of molecules on sub-diffraction scales on something that is smaller than the size of the virus. And here we applied so-called scanning state fluorescence coloration spectroscopy to get the details, as you can see on the left, to get the details of how the molecules move across an individual virus surface. So this is actually on the left an image of several virus particles labeled in green, and then we studied them using the STEAD-FCS technique that I mentioned, and we acquired correlation times and molecular mobility data on each one of those individual virus particles and its NF mobility. And we actually found that on the mature virus particles, we found that first of all the virus environment is very, very slow. The molecules are moving very, very slowly, much, much slower, an order of magnitude slower than what would be expected on a cell plasma membrane. However, even having said that, on immature virus particles, the molecules of NF were moving even slower than on a mature virus particle. So there was a quite significant difference. And of course, on the fixed virus particles, everything was immobile. So it showed us that the study is well controlled. We found out that actually HIV envelope is intrinsically very immobile. This is actually irrespective of NF. When we looked at the cells which have been labeled with a GPI anchor, which is a very tiny molecule compared to NF, we found average transit time to be consistent with what the molecules of this size are supposed to be. But when looking at the virus that is mature or immature, we found out that even those small molecules slowed down an order of magnitude on the virus surface. So basically, this told us a lot about and how HIV environment is completely different to what you expect on what occurs on a cell surface, for example. So the conclusions from this study that HIV has a very low surface mobility, and that is at least order of magnitude smaller than on the cell membranes. That virus maturation actually influences end mobility. But the bottom line was from this study was that HIV is incredibly low mobility environment for the surface molecules like ENV. And this applies to other molecules that also hitch a ride on the surface of the virus. But I guess this is understandable because normally when we look at the cartoons of HIV, we think, oh, look, there is just some molecules and it all looks fairly empty on the inside. But in reality, when you look at actually true molecular structure of HIV, it's an incredibly packed, complex system. It's full of molecules which are interacting with one another, and therefore it's no surprise that a molecule with such, as in a system of such curvature has a very high lipid packing, high protein packing. The molecules move so much slower than on the surface of a cell, for example. And then going to the final study, which I want to mention, we then looked at lipid and end mobility at fully infectious HIV assembly sites. So, a little bit of a background. HIV membrane is highly enriched in sphingomyelin and cholesterol, but the consensus was that virus assembly actually occurs on so-called lipid rafts, which are cholesterol-enriched domains which are pre-existing on a cell surface. But here we wanted to test a hypothesis that maybe instead of targeting pre-existing lipid raft domains, the virus actually creates its own bed for assembly and creates its own unique environment for virus assembly. And we did that using also fluorescence coloration spectroscopy, but this time we went into full live cell imaging where we focused on individual virus assembly sites, as you can see on an infected Jurkat T cells in 37 degrees. And then we actually did a fluorescence coloration spectroscopy on areas of the virus assembly site, inside and outside virus assembly. And therefore, we were able to gain information not only what happens at the virus assembly site, but also just outside of it, and be able to compare the lipid environment, lipid and protein environment inside a virus assembly site and just outside of it. So, just to cut to the short, we found out that it was the PIP2, which is an important molecule for HIV assembly, and cholesterol are displaying reduced mobility at HIV assembly sites, but not just outside of assembly site, and of course not in uninfected cells, but molecules like sphingomyelin or phosphatidylethanolamin, which are more neutral lipids, do not seem to get trapped. And of course, in addition, ENV molecules also become hugely trapped on the surface of at the point of virus assembly. So basically, this told us that HIV, actually, contrary to what was thought before, is able to create its own very specific lipid environment by selectively restricting some lipids at virus assembly site. And this environment, combined with the immature Gag-lattice interaction, leads to this end trapping and clustering, which was I've shown in the previous studies. So this is, of course, a very brief snapshot of quite a long, about 10 years of research. But I want to basically leave you with a basically take-home message that super-resolution microscopy is a very powerful tool for studying HIV and also, of course, other pathogens, and it has potential for making high-impact discoveries. And right now, with where the field is, with the current availability of commercial microscopes being much greater than a few years ago, these techniques can actually be much more easily applied by groups, by a larger number of groups, research groups than before. The field is no longer constrained to a few groups which have dealt with this field from the beginning. Now the field, I would argue, is very open to everyone who has access to the microscopy facility. Of course, still you need still a guidance. But like I said, those tools are now available to a larger amount of facilities and available to more research groups. And I'll finish here with just a little bit of the advertisement. We actually right now in process of finalizing one such platform at the Canruti campus in Barcelona, where such super-resolution microscope will be available as part of collaboration, as part of a service, if anyone wants, in a P3 environment, which allows you to study fully infectious virus particles. So here I'll stop, and I will I would like to first thank the group of Javier Martinez Picado for hosting me for all these years at the group. Then, of course, all the thanks go to the previous groups that I have worked with to show you all of those studies. So with that, I'll stop and take any questions. Thank you very much.
Thank you, Jacob. If there is any question, please, this is the moment. Yes.
I'll ask in English, right?
Sure, please. English a little bit better.
Thank you very much. This was wonderful. So I was wondering if you're considering or you have started working towards the direction of looking at virus produced in myeloid versus T cells. Because, you know, we have been struggling, not us specifically, but in general, the field, struggling with techniques like flow virometry or lipid fingerprinting. So this sounds like it would be a lot simpler to just look at the variants themselves and try to see if there is a different pattern of the lipid structure of the lipids or the mobility maybe. I don't know.
Yeah, the answer is this was floated around before. I just never got around to it. You know, I focused on, but that definitely the myeloid with its T cells and the differences in virus. You know, definitely we thought about that at that time. I just never got around to it. So if anyone wants also to collaborate or do study like that, I'm very, very open. You know, my expertise is you know, I'm happy to collaborate on that. But it was just simply due to the lack of time to do that sort of thing. But certainly, exactly, being able to look at the structural differences on molecular level, look at the distribution of ENV, look at the lipid profiles, these techniques precisely would allow you to do it in live context, which I think is very relevant here.
Any more questions?
Hi, good morning. Jose Martinez from University of Miami. Great talk.
Thank you.
Mind-blowing, actually. I wanted to ask you, you know, if the breathing of the envelope, they say that it breathes between the open and closed conformation, would make any influence in your motility assay? And second, if the number of envelope per particle will also make any influence, or if you tested that and saw, you know, many envelopes will move slower or the like.
Well, first of all, it's hard to say exactly opening and closing. We didn't test that. We looked at, you know, this was all antibody bound. We did not tag the ENV structure directly because that's difficult. But we looked at antibody. Basically, we look at the region which is very conserved. We looked at 2G12 antibody, which is basically very conserved region. True, we did not look at that, how open or close ENV structure may make a difference. But I think the most crucial determinant of the mobility is its interaction with the lipid membrane. I think that the anchoring of transmembrane domain and also the long cytoplasmic tail is what makes the ENV so slow. Probably less so the basic external structure. And can you please remind me the second question? Sorry.
The second question one is if the number of envelopes would make an influence in the motility.
We also quantitated, these techniques are also quantitative. We quantitated the amount of ENV molecules, and we also got to the number that has been previously quoted by cryo-electron tomography, by cryo-EM. We got around 7 molecules. The number of ENV molecules would make a difference only if you start getting a whole lot of molecules, which would make it more difficult to distinguish individual clusters. But of course, the system that we have utilized here, I think will quite nicely show that it recapitulates what was shown by electron microscopy. So we actually didn't test, you know, a situation where we loaded the virus with a lot of more envelopes. This was something that we actually honestly didn't do. We were happy that we had a system that, you know, that has been shown to be consistent with the data with cryo-EM, for example. Sorry, electron microscopy. So
it looks pretty cool. Thank you.
I have a follow-up question regarding that, because all these studies have been done in cell lines or in primary cells. So
at the moment, this was all cell lines. It's true. The viruses that were grown is 293 cells. Exactly. There is, of course, a lot more that you can do. You can start looking at the primary cell-derived virus. You can look at the founder viruses and so on. And all these projects were one at a time floated to me. As I was, for example, leaving Oxford, I just simply had to say, sorry, I just don't have time. I'm going somewhere else. So there is definitely room for more. So this is exactly the point of my talk. There is, you know, the techniques are now available, and if you have those questions, it's no longer confined to a very small amount of researchers which have the know-how. The techniques are there, and the studies can be done. So it'd be exactly nice to have it, more and more groups contribute to the field.
And it's great that you have it in these health-free facilities.
Exactly, so. it's here exactly just around the corner, so to speak.
Thank you. Amazing talk, really. And I have been working, so my name is Eloisa Juste. I work at Instituto de Salud Carlos III, but I have a past working with envelopes and spikes and truncating the cytoplasmic tail and all these things. So I'm assuming that everything you are talking about is done with a full cytoplasmic tail.
Yes, we've done the study on the both full cytoplasmic tail, but also on the delta CT variant, which cuts off the tail, and that was found that the cytoplasmic tail has a very, very big effect on the clustering,
yeah,
and also on the mobility of and it actually does act as a strong anchor and slows HIV env down. And to get to a bit more details, we found out that actually lack of the delta CT, lack of the cytoplasmic tail was what rescued the HIV delta CT mutant infectivity, because we found that when a virus is free from the constraint of the, virus N is free from the constraint of the delta CT, the cytoplasmic tail, the molecules can recluster and at least recapitulate partially the infectivity of this virus, because in the field before this study, it was a bit of a puzzle. How does delta CT mutant recover its infectivity?
So how having the truncated cytoplasmic tail is affecting the mobility of the spikes in mature and immature virions? Because
in mature and immature, we found that it does not have any actual difference. It's all about 200 milliseconds transition time, but it does affect the clustering. That's actually to do mostly with the underlying gag lattice. So it's all about the connection to the gag lattice. So if this immature gag lattice is very rigid and highly structured, then the ENV is anchored via C-terminal tail to the lattice. But as soon as the lattice disassembles, the ENV is now more free to move to coalesce into the clusters. But if we look at the immature and mature virus particle in a wild-type ENV, you can see that in mature, the ENV is twice as slow or almost immobile. This is actually a limit almost of the technique. But it speeds up just a little bit when we have the fully mature virus particle. So that is basically what once the virus ENV is released when the gag lattice disassembles. But it's still very, very slow compared to what you would expect on the cell plasma membrane, ENV on the cell plasma membrane, for example.
And have you done these studies with SIV?
Sorry, with what?
With SIV?
No.
No?
The thing with SIV is that SIV actually has paradoxically quite a lot more ENVs on the surface. I think
only when it's truncated.
Sure, true. But in the end, that's why we focus on HIV, because precisely at a time, this was early studies of superposition microscopy, we're very happy to have only a few molecules of ENV, because as you can imagine, if you have more molecules of ENV, then you still, at some point, you will lose the clusters and you will just get a ring around like for example, if you image SARS-CoV-2, you probably would get a ring around, as an image ring of all the spikes around, and you would not be able to look at clusters.
Yeah, but if you have a wild-type SIV, you also have 7 spikes.
Sure,
so you can do the same.
That we just simply didn't get around to. We at a time exactly. We wanted to look at. It's true. In this case, it was simply we focused on HIV here.
Here, aquí delante, por favor.
Thank you. Very nice presentation. I was just wondering how the different distribution of the envelope molecules in immature versus mature virions affects the neutralization of the antibodies.
The localization itself, to be honest, I don't know. But there was a follow-up study, which looked at the EMPER region, and I think if I recall correctly, I don't think there was found any difference between the mature and immature. But this is something that I simply don't know if there will be anything. There is some literature on this, but I cannot find. I don't know at the moment. Sorry.
Okay. Thank you.
¿Alguna pregunta más?
Okay, I have a question. Sure. So, thank you very much. The talk is impressive. So I was wondering, so at this resolution level, so can you take a look to the beyond the envelope of beyond the surface of the cell and go into the escort machinery, for example, to take a look to how HIV assembly at a molecular level in the machinery that is involved in HIV assembly?
Yes, you can. I mean, right now, I mean, the post-entry stages, disassembly as well. Originally, assembly and entry was the first thing that studied because it's relatively easy to on the surface, and you can study surface molecules easier. But right now, exactly, thanks to actually advances in labeling and click chemistry, for example, really enable to study all those more advanced and deeper steps of HIV replication cycle and combine it with the superposition microscopy. So yes, group in Heidelberg, my former boss actually, he's actually made a headway into it, and they were one of the first ones to study post-entry stages, the capsid uncoating, and actually were the ones to found this the nuclear disposition of the capsid, but also additionally, looking at the perinuclear stages of virus replication cycle. So yes, right now you can, since the field once again has advanced so much, you can study all these not easy parts of HIV replication cycle, definitely. And of course, transinfection, as in synapse as well, and so on. It's all possible right now.
Okay, thank you. That's very interesting. Any more questions?
Thank you.
Okay, thank you very much. It's been a terrific talk. Yes.